Hello, my name is Ayaan Arif, and I'm going to be presenting my review paper on the role of HAP1 in the progression of Huntington's disease in middle-aged patients. So I'm just going to start this off with an introduction to Huntington's disease, commonly abbreviated as HD. So Huntington's is a rare adult onset genetic neurodegenerative disease. A neurodegenerative disease is a type of disease that targets the nervous system and slowly breaks it down over time. So it'll affect different areas, and examples of diseases like this are Alzheimer's. And a lot of these diseases are always fatal, and Huntington's disease is one of these that are always fatal. So it is inherited genetically and is autosomal dominant, but is more common in females than males, but there's no particular mechanism that explains why this is. So due to being adult onset disease, you only see symptoms start at around 40 to 50 years old, none before that. However, you can test, but you won't see any symptoms. And it is malignant, which means that symptoms worsen over generations, and there's an earlier onset in successive generations. You can have a juvenile onset at less than 21 years old if the disease passes on over time. And the hypothesized cause for Huntington's is an expanded CAG repeat in the Huntington gene. And it is expressed cytoplasmically, so you'll see it in the cytoplasm, and you'll also see the cells also die over time. But a lot of the main disease pathways, like the brain region first affected, are unknown. So the genetic basis of HD is basically within the HTT gene, the Huntington gene. And it's located on chromosome 4 and it provides instructions for the Huntington protein. So the gene is shown on the right, that big strand. And what basically is the cause, as I said earlier, is a CAG repeat number. Healthy individuals are seen to have less than 26 repeats, while affected individuals have less than 40 repeats within the gene. So the CAG repeat is present within the HTT gene, just to make sure. And the function of Huntington is currently unknown. The protein is found everywhere within your body, but it is highly concentrated within your brain, your testes, and your gut. But it is theorized to be involved in chemical signaling, but when mutated, it causes degeneration and affects voluntary movement regions like the cerebellum and basal ganglia. So I want to talk about two main structures, HAP1, Huntington's associated protein 1, and FANC1, FANC1 associated nuclease. HAP1 is a neuronally expressed protein that is crucial for the survival of a neuron, and its localization within the body is similar in monkeys and humans, but different in rodents, which will be important later. And FAN1 is shown to stabilize CAG repeats and slow down HD progression as well. So HAP1 and FAN1's interaction with HTT, a possible connection can be formed between all of that, and that connection may affect HD onset and its severity. And a little tidbit of information is that when FAN1 is knocked out, HAP1 has a higher binding affinity to mutated Huntington. So here's my research hypothesis. HAP1 interaction with mutated HTT triggers the onset of HD. Methodology of how I would do this would be to analyze literature on HD, HAP1, and FAN1 due to me being a review paper, and I'd compare these findings to support my hypothesis. So right here is a flowchart I made showing how I got my hypothesis. So I used PubMed to perform a quick literature skim on Huntington's using similar search terms and whatnot, and I narrowed down the multitude of papers I found into five main papers that basically I would quote throughout my paper and would provide me a lot of evidence. And the common ground between them was that a lack of HAP1 would lead to high CAG repeats and Huntington's disease starting. So this led me to the conclusion that HAP1 is a main protein that prevents HD, and without it, HD actually starts. So here's the start of my research findings. HTT and HAP1 expression in similar brains. So HTT is expressed in all brain regions and cell types within the brain, and differential expression can be seen within monkeys and humans. So just to bring your attention to the figure on the left, you can see that top row is mouse, only mice. So as I said earlier, there's different expression between rodents and primates. So you can see where HTT is present, marked in red, is not the same as HAP1. You can see those regions are different. However, in the bottom, where they're monkey, HTT and HAP1, wherever the HTT is present, HAP1 is also present. So basically what you can draw from this is that similar expressions in two organisms suggest similar progressions of HD. which is true because we know that monkeys and humans have similar HD progression while mice do not have as close a similarity. Different HAP1 expression in rodents and different onsets together suggest a different and a higher role of HAP1 in the onset of HD. So here's FAN1 and HD progression. So just a quick study overview of the information I used. A TWAS was taken on 452 HD patients, and that basically just checked which specific genes altered the age of onset of HD. So the age of onset is basically just the age at which you see the symptoms start. And you can see a correlation between FAN1 levels and HD onset. So basically the orange that is lacked that is marked as cells with FAN1 while blue is without. So we know that FAN1 is basically just controlling CAG levels. So you can see that the blue ones over time the CAG level increases while with the orange one it's more stable increase. So basically what that proves is that higher FAN1 levels equals slower HD progression, while lower FAN1 levels equals an earlier onset, which could also be a cause for the juvenile onsets and malignancy. So basically all that proved was that, oh, FAN1 does slow down HD progression and is very important. And there was also another study that examined postmortem samples of humans. And it basically cross-referenced this study and it basically showed that same results are true. Higher CAG repeats are linked to faster HD onset, while FAN1 does stabilize those repeats, but its absence causes instability. So moving on, I would like to talk about some calcium release. HAP1 HTT and INSP3R1, which is a calcium release channel, their interaction altogether greatly increases Ca2 plus release. And an excessive Ca2 plus presence within a cell is linked to early HD symptoms and cell death. Cell death is one of the early HD symptoms. And I just want to bring up the fact that HAP1 is not necessary for altering the Ca2 plus levels in the basal ganglia. And it's also not required for the interaction between HTT and INSP3R1 if the CAG is long enough. So HAP1 isn't as present as HTT, but it can still lead to Ca2 plus releases, which leads to the cell death, which can be associated with HD. And all this information basically shows that this might be the cause for neuronal death associated with HD. However, this information is not truly concrete and it is a lot of speculation. So just this brings me to my discussion and my conclusion. So most evidence suggests that HD stems from mutations in proteins like FAN1 and HAP1 and HTT and the production of the mutated Huntington. And the altered levels of these proteins can lead to changes in CAG repeats within the HTT gene, causing HD symptoms like cell death, which I mentioned earlier. So HAP1's role in a study I showed earlier within the monkey brains, it demonstrated that HAP1 is essential for maintaining CAG repeats in HTT along with FAN1, and the knockout of HAP1 leads to an increase in CAG repeats, implicating HAP1 as a potential HD-causing gene when mutated or knocked out. So FAN1's role leads to a slower HD onset, and it is associated with a slower onset as well, suggesting a protective mechanism involving FAN1 and HAP1. And here's some further implications and research we can do. HTT and calcium release. So the study with basically INSP3R1 and the calcium release showed that mutated HTT binds to INSP3R1, which causes excessive calcium release and cell death within the brain, which is associated with the HD symptoms. And the elevated Ca2 plus levels are early sign of HD onset, which brought me to the conclusion that it's the main symptom or the main cause for the symptoms of HD in the beginning. And my main conclusion is HAP1 appears to be the most crucial protein in preventing HD. While further research could include adding HAP1 to the CNS to bound CAG repeats, but we do need further technological advancements. And here are my references. And thank you for listening to my presentation.